Hi everyone, this is Ulrika from the Watson Lab at the Physiology and Pharmacology Department. Today I'm here to talk to you about the effects of palmitic acid and oleic acid on preimplantation embryo autophagy. So last year, the World Health Organization published data saying that they estimated 52% of adults worldwide were considered overweight or obese in 2016. So to bring things a little closer to home, Statistics Canada also published in 2018 that 63% of Canadian adults were overweight or obese. And within that population, 57% of them are females. Many of these females are of reproductive age and are at the family planning stage of their lives. However, many of them actually experience fertility problems. Obesity has been found to be evident in reducing fertility potential, in which women who have high BMI levels would experience disrupted gonadotropin production, menstrual abnormalities, and ovulatory dysfunction, as well as a higher incident rate of pregnancy complications. And so to serve this population of women who seeks help for their fertility needs, our group investigates the effects of non-esterified fatty acids, also known as the NEFAs, on preimplantation embryo development. The embryos spend their preimplantation stages in the fallopian tubes, and that they're highly affected by the surrounding environment, which is the follicular fluid. The profiling of the human follicular fluid has found the presence of NEFAs. Similar to the human serum levels, palmitic acid, PA, has found to be the most abundant saturated fatty acid, while oleic acid, OA, is the most abundant unsaturated fatty acids in the follicular fluid. So our lab focuses on these two types of fatty acids, and recently we discovered that PA would induce ER stress and impairs blastocyst development. Meanwhile, OA would increase lipid droplet accumulation and decreases mitochondrial stress. Additionally, we found that OA can counteract the effects brought upon by PA treatment. Now, all of these pathways that we looked into would eventually lead to apoptosis under high stress conditions. However, there is a protective mechanism that the mammalian cells would use before initiating apoptosis, and this mechanism is known as autophagy. Autophagy is a survival mechanism in which to protect the cell from severe cell stress. So this mechanism enables the formation of autophagosomes so that the cytosolic components that are not of use anymore can be packaged and then eventually fused with a lysosome later on to be degraded into metabolic precursors. So these degraded contents will then be released back in the cytosol for reuse in other cellular functions. So autophagy has been found to be essential in embryo development, in which previous studies have found that inhibition of autophagy by knocking out one of the very important autophagy-related proteins, ATG5, would lead to developmental arrest at the 4-cell stage and even lead to embryo lethality. It is unknown whether NEFA exposure in preimplantation development would have an effect on autophagic mechanism, and so it leads to our hypothesis that PA will alter autophagy during early embryo and embryonic development, and that OA would reverse these effects. To look into this further, we have CD1 female mice that are gonadotropin superovulated and then mated. Mouse embryos were then flushed at the 2-cell stage and then incubated with the caseum growth media, 100 micromolar of PA, 250 micromolar of OA, and 100 micromolar of PA and 250 micromolar of OA in combination for up to 48 hours. So after the treatment period, the embryos were collected for developmental stage analysis and then used immunofluorescence staining for autophagy marker expression, as well as RT-qPCR for transcript analysis. So first of all, we were able to confirm that in fact, PA treatment for 48 hours does impair blastocyst development. So we are able to identify that PA treatment would have results in developmental arrest at the 4-cell and the 8-cell stages. So PA treatment in 48 hours results in significantly lower blastocyst development, and it's evident that embryos were significantly arrested at the 8-cell stage when compared to other treatment groups. So to look into this further, we looked into the changes in autophagic marker at the transcript level. So we found that the autophagy markers for autophagosome formation, which we looked into Beclin-1, ATG3, ATG5, as well as LC3, and these markers were all unaffected by any of a treatment at the transcript levels after 48 hours of treatment. However, when we looked into the protein expression of autophagic markers, we found that in fact, PA does alter autophagy in mouse embryonic development. So we looked into activated LC3, also known as the LC3-2 puncta, and it is a marker for the presence of autophagosomes. So these embryos were treated for up to 48 hours, and that, as you can see, the embryos treated with the PA group has the higher amount of LC3-2 aggregates compared to other treatment groups. So we were able to identify that LC3-2 expression levels were unaffected by any of the treatments at the early stages of 18 and 24 hours of treatment. However, after 30 hours of exposure, both PA and the PA-OA combination group have a significantly higher LC3-2 expression when compared to the OA group. Now, this effect is extended to the 40-hour time point, at which both PA and the PA-OA combination group would result in a significantly higher LC3-2 expression than control. So at the 48-hour time point, it was found that the PA group significantly increased expression of LC3-2 compared to the OA alone and as well as the PA-OA combination group. So this is an indication that the addition of OA in PA treatment media would significantly reduce LC3-2 expression, but only after 48 hours of treatment. Last but not least, it is important to note that OA treatment group is not different than control on LC3-2 expression throughout the entire period of mouse preimplantation development. So we were able to identify that PA does in fact impair blastocyst development by arresting embryos at the 8 cell stage. And it also results in significant changes in autophagic marker expression of LC3-2 aggregates. However, these effects were successfully reversed by the addition of OA in the treatment media. So with these discoveries, we were able to understand the effects of NEFA exposure on autophagic mechanisms, as well as to highlight the importance of autophagy in preimplantation development. So here we present the first indication that NEFA exposure affects autophagy in early embryonic development. We believe that altered autophagy may impact developmental competency of early embryos, especially those that are derived from obese patients. So our next step is to measure autophagic flux of NEFA-treated embryos as to identify for the precise effects of fatty acids in early embryo autophagy. We will also look into the therapeutic approaches to alleviating negative impacts brought upon by hyperlipidemia during early embryonic development. We would ultimately like to enable fertility success for overweight and obese patients who require help in their fertility needs. So lastly, I want to take this time to thank our funding sources, the Canadian Institute of Health Research, as well as the Children's Health Foundation. Also, I want to thank our Beth Watson Lab members and my advisory committee members for the support throughout the project. Thank you.